Lab number 8 has 4 parts to it. In this lab, we are looking at different patterns of human inheritance. So we're going to look at pedigrees, we're going to look at sex linkage, blood typing, and human genotypes. For each of these, I have probably already done a practice problem or two, and those should be available in other places on Blackboard, either in most likely in the Chapter 9 folder, possibly in the Chapter 9 assignment folder. Let's just do a quick review of pedigrees. So the 3 main types of pedigrees that you're going to see are autosomal recessive, autosomal dominant, and sex-linked recessive. You can also have sex-linked dominant pedigrees, but they're a little bit hard to decipher from autosomal dominant. And so for this course, I very likely will not give you a sex-linked dominant. So we're not going to worry about those too much. So there's a couple ways that you can recognize the various inheritance patterns in these pedigrees. And then it's always a good idea to confirm them by adding in the genotypes, just to make sure that your genotypes make sense, to make sure that there aren't children inheriting alleles that they can't inherit from their parents. So let's look at autosomal recessive. The key to an autosomal recessive is seen right here in these individuals in the middle. In an autosomal recessive, you have 2 unaffected parents and an affected child. Remember that the filled in squares or circles mean that the individual is affected with the trait. Now, affected doesn't always mean something negative. It doesn't always mean a disease. It might simply mean that they have a particular hair color or a particular physical feature. But when we look at autosomal recessive, the one thing that we can always know for sure indicates an autosomal recessive is these 2 unaffected parents with an affected child. That means that those 2 parents are heterozygous individuals who are hiding the allele and passing it on to their affected child. So they're carriers. So I always look for that particular trait of a pedigree when I am starting out, because that's the easiest one to identify, and it's the only way that you can produce this pattern is if that trait is recessive. The next autosomal type of inheritance that you might see is autosomal dominant. Now, in autosomal dominant, you're going to look for every affected child having an affected parent. Now, the reason is that you cannot hide a dominant in a carrier. The dominant is always displayed if you have that allele. So if we start down here in the last generation, we have an affected child, an affected parent. These 3 females are affected children with an affected parent, affected child, affected parent, and so on, all the way up to the top. Now, the other way that you can sort of think about this is that it appears in every generation. But what that means is that it appears in every parent-to-child generation. So we have parent-child, 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 etc. Not it appears in this generation and this generation and this generation. And what I'm trying to explain is that if you look over here on the recessive, it appears in this generation, and it appears in this generation, and it appears in this generation. But we want to look at the direct line. And if you look at the direct line, parent, child, child, you don't see that inheritance in every single generation. So just sort of keep that in mind. That's what we mean by appears in every generation. It means that every parent to child shows that trait. So then our last one is sex-linked recessive. So the sex-linked recessive is going to be mostly or exclusively seen in males. So if we look at this pedigree here, we have all affected individuals are males. We have no affected females. Now this one gives us a little hint that's really, really helpful, and you won't probably see this if I show this on an exam. But this one also shows carrier females. So you'll notice that females are carriers and pass it on to their sons. That's how you know that this is a sex-linked recessive. But even if you're not shown females being carriers, the fact that all of the affected individuals are male tells you that this is sex-linked recessive. Now, in a pedigree of this size, it might not be uncommon to see a 1 or 2 affected females. But if you have, say, close to 50% affected females, you definitely do not have a sex-linked recessive. In a sex-linked recessive, the majority of the affected individuals are going to be males. So our lab also asks us to look for and write in the genotypes. So I'm not going to do all of these, but I'll do a couple. This male here would be hemizygous recessive. That means they have only one X chromosome and it's recessive. The male in the middle here would would have the dominant allele there, and you know he has the dominant because there's only one allele and he's not affected. This one would be recessive. This female would have to be heterozygous because she does not have the disorder, but she does pass it on to her child. This female could be either homozygous or heterozygous. We wouldn't know for sure until we saw her children. This female here, we know that she is heterozygous because she passes it on to her child, and dad is going to have the dominant trait there. If we go up to this autosomal dominant here, we know that anybody who does not have the trait is going to be homozygous recessive, and anybody that does is going to be either heterozygous or homozygous, but you have to determine that based on their children. So for example, this couple here has a child that's homozygous recessive, so we know that dad has to pass on that homozygous allele. Over here, anybody that is affected is going to be little a, homozygous recessive. And again, all of the others are going to be either homozygous dominant or heterozygous depending on the children. Again, because this mom produces an affected child, we know that she has to be heterozygous because each parent has to pass on a specific allele. Over here, let's look at these individuals. This individual is affected. This individual could be big A, or big A, little a. But we don't know because they don't have any affected children. We don't know for sure if she has that recessive allele. So same thing with this generation here. Both of these individuals could be either heterozygous or homozygous, and we wouldn't know until we looked at their children. In exercise 8.2, we are looking at sex-linked inheritance. In order to work out the problems here, you may want to review the sex-linked inheritance video that I posted on Blackboard. It's probably under the Chapter 9 assignment or under the Chapter 9 lecture videos. And there's one specific video on how to do sex-linked problems. But for this one, we are looking at eye color in fruit flies. Eye color is a sex-linked trait, and the white eye color is recessive to the red eye color. So for this section of the lab, you'll need to refer to the Blackboard posted data, and you're going to look at a set of flies, and you're going to look at their physical characteristics, abdomen size, the presence of sex combs on their legs, and then of course the color of their eyes. So read carefully through the lab manual. It explains how to determine males versus females. And then look very carefully at those flies. They're small, I understand. They are hard to tell, but do your best to put them into male-female categories. And then usually the eyes are pretty easy to see. The red is pretty bright, and if you don't see that bright red, it's probably a white eye color. So from there, you're going to start out with a cross, and the flies in the images are a cross between a red-eyed female and a red-eyed male. So the first thing you're going to need to do is to determine the genotypes of these individuals. Now, this is a sex-linked trait. Remember that males have only one allele on their X chromosome, and if the male is red-eyed, which is dominant, they're going to start out X big R Y, and we're going to be crossing with a red-eyed female. So we know that she has at least one red-eyed allele. You're going to have to look at the progeny or the children of this cross to determine if this female is heterozygous or homozygous. So you'll have to fill in their genotypes. And then you're going to count up the number of individuals and determine whether or not it fits the expected ratio from the given cross. In section 8.3, we're looking at human blood groups. So you're probably already familiar with the different blood groups. We have type A blood, type B, type AB, and type O. Now these blood types relate to the antigens that are present on the outside of the red blood cell. So an antigen is simply a protein or a carbohydrate that elicits an immune response, basically. So the A blood group produces an A antigen. So this is just a special carbohydrate that's on the outside of the blood that identifies it as a blood type. B blood type has a different type of antigen that signals that it is a type B blood cell. Type AB blood cell has both of those antigens. That's why we call it AB. It's got the A antigen and the B antigen. And then type O blood has no antigens on the outside. So this is important because when they have antigens, this can elicit an immune response because of antibodies. So if you are an individual with type O blood, you produce both A and B antibodies, which means that your blood will produce a reaction in the presence of those A and B antigens. An individual who has type A blood is going to produce B antibodies. So B antibodies. And if you look here, you can see that these antibodies could recognize these triangular shaped B blood cells. The type B blood is going to produce A antibodies. And you might notice that they would recognize these A antigens. Now this means if you have type B blood and you are given type A blood, you are going to make an immune response against that blood type. This is a bad thing. If you are type A blood, you have both antigens, which means you actually don't make either antibody, which is kind of cool. So you can accept any blood type. Blood type O, because it doesn't have any antigens, can be a donor. They can donate. Individuals with blood type can donate to anybody. I realized that I just misspoke, so I went back and deleted. If you are type AB blood, you can receive any blood type, and that's because you don't produce any antibodies. If you are type O blood type, you can donate to anybody, and that's because you do not have any antigens. So let's move on. We are going to ask you to do some blood typing here. Now, we've provided you with pictures in the data file for these blood typing reactions. So essentially what you are doing is you are attempting to make a clotting reaction. You are attempting to make an antibody reaction. And so if you add the A antibody to a type A and you get a reaction, if you have clotting, well, A and RH have clotting, that means that that is the blood type that you have. So if you get a clotting reaction for any of these, that means that you have that blood type. So if you get a clotting reaction for A, you are blood type A. If you get a clotting reaction for RH, that means you are RH positive. No reaction here for B means you do not have any B blood type. Now, if you were to have a reaction here, you would either be AB if you also have an A reaction, or just B if you only have a B reaction. If there's no reaction on the RH, that means that you are RH negative. So you'll look at each of these, decide or look to see what has been added. If you are anti-A and you have a reaction, then you have type A blood. If you are anti-B, or sorry, you add anti-B and you have a reaction, you are type B blood. And then finally, for exercise 8.4, we're just asking, what is your genotype? So we give you these different traits. We ask you to look at yourself in the mirror or look at your particular appendage, whatever it might be, and decide if you have the dominant trait, the recessive trait, and then give us your potential genotypes. Keep in mind that if you have the dominant genotype, you could be homozygous or heterozygous. So if that's the case, you need to indicate that it could be either one. Pay attention, because at least one of these is a sex-linked trait, which means you have to have the sex-linked notation to go along with it.